So our poster is about identifying novel variants in ABCC6 and other modifier genes in PXE patients. So PXE is basically a genetic disease. It's autosomal recessive, and its main symptom is the calcification of the dermis. So the early symptoms are papules on the neck of PXE patients, like you see here. And when you do a biopsy and stain it with von Cossa stain, you can see these red stained places, which are where calcium deposits can be found. Another phenotype of PXE is cracks in Bruch's membrane, which is behind the retina, which leads to angioid streaks or blood vessels growing through the retina. So ABCC6 is an ABC transporter. It has 17 transmembrane helices and 2 nucleotide binding domains that you see here. And most of the mutations in the patients are in the nucleotide binding domains, which kind of makes sense because these are the functional domains responsible for ATP binding and hydrolysis and ultimately for the transport of the substrate. So the way we did the project was that we took DNA from PXE patients and amplified them on a fluidigm array. So this is basically a glorified PCR. And then we sequenced them on a 454GS Junior sequencer. And then after sequencing, we got an Excel table of the different variations in the ABCC6 gene and in the 13 modifier genes. And what we did then was we validated the next-gen sequencing, which is very high throughput and therefore not as accurate as Sanger sequencing. So we validated the results we got from next-gen sequencing using Sanger sequencing in order to discover novel variants and mutations. So if you look at the table here, we discovered around 10 novel variants, or we thought we discovered, but some of them were false positive, and in the end what we found was 2 novel variants in ABCC6 after validating by Sanger sequencing. These are G750S and L753P, and both of these are found in the nucleotide binding domain. So here is G750S and L753P in NBD1, and these are just the Sanger sequencing traces showing the 2 mutations. Another thing that we did was we wanted to see if these mutations are deleterious mutations, so we ran them through Panther. And what we see here is the sub-PSEC scores on both of the mutations are lower than -3, which basically suggests that these mutations are harmful mutations. So we actually sequenced around 42 patients. 24 of these patients had been screened before. So here we see the 24 patients who had been screened before by PXE International, and you can see in most of the patients they couldn't detect the mutations, and all these patients actually had PXE, but the mutations were undetectable. However, when we resequenced these patients using next-gen sequencing followed by Sanger sequencing, we actually detected most of the mutations except the large deletions, which cannot be detected by next-gen sequencing. Finally, we also sequenced all the exons of the 13 modifier genes, and we actually found SNPs that could be associated with PXE. So we found these SNPs in 8 of the modifier genes, and we are now currently validating them using Sanger sequencing in the laboratory. So in conclusion, we can say that we discovered two novel mutations in PXE patients, L753P and G750S, both of which are in the nucleotide binding domain, and both of them are predicted to be deleterious mutations. We also showed that targeted exome sequencing on a 454 platform can be used to rescreen patient DNA to detect previously undetected mutations. And we also discovered modifier gene variants and single nucleotide polymorphisms in PXE patients, which actually might correlate to the phenotype of PXE patients, but we're still working on this right now. And our future directions are basically to expand this study to a larger cohort of at least 100 patients and 150 controls to determine if these SNPs and modifier genes associate with particular phenotypes in PXE patients, and also to detect more novel variants in PXE patients.